right. Thank you very much.
Yes,
To set the ball rolling for questions to come in, let me start so that you can throw more light on your presentation. So first of all, let me just give a brief summary. You have introduced biological databases. You told us the importance. You told us the types. And then you also mentioned some important websites. Now, some key terms. Please explain to us what we mean by ligands, proteins, and targets, and the relationship among these three. Ligands, proteins, and targets. And what relationship exists among these?
all right, thank you so much for the question. So proteins, ligands, targets. So I'll just use the starting points will be probably like there's a disease you want to treat. If I'll still use the example of diabetes that I was using before. We know how amylase helps in like glucose breakdown and the likes. So alpha amylase is an enzyme and is a protein. So that's what proteins are. We like generally we know what protein does. They help in transportation in the system. They also act as enzyme to speed up the rate of chemical reaction in the system. And then if we want to look at probably metabolisms of glucose, we know that in that glycolytic pathway, there are lots of enzymes, and these enzymes are protein. So it could be probably the rate limiting step enzyme that you want to target, but I will use alpha amylase as my example. That's an example of protein. So your target could be alpha amylase, which is a protein that performs an enzymatic function in the system. And if probably the activity is reduced or increased, like above the homeostasis level, it could lead to disease states. So if you're talking about diabetes, we know it's a whole lot of complex reactions. So, but I just want to relate that to what's protein, and I'm trying to like give an example. So if your target is alpha amylase, you want to probably enhance the activity of alpha amylase. So first thing is you want to start from probably the form of treatment that have been available before, and then the drawback in this treatment. So that's where ligands comes in. Ligands are like small molecules that could bind to a protein. So a protein could be the target, a gene could be the target. It depends on what you actually want to target to either inhibit the activity of your target or to enhance the activity of your target. So it depends on what you actually want to do. And this can be done. I mean, you can actually have a hands on what you want to do by studying the literatures and then know, okay, where science is in particular. So they know the biological pathways that are involved in diabetes. So before you could know where to come in, probably what your target should be. So if you study like the metformin, which is a drug that has been used or still in use in the treatment of diabetes, you could read the bioactivity and how it interacts, what the target of metformin is. And if you want to improve on this drug, so the drug could be the ligands, which is like the small molecule that affects the activity of your targets or a protein or another big or biomolecule. So that's what ligands are. They affect the activity of a biomolecule or of a big molecule in quotes. So ligands are like small molecule. So once they attach to, probably it could be the active site, it could be the allosteric site of your protein, will affect the activity of the protein. So I hope like that has been able to answer. So your target could be a gene, it could be a protein, it could be whatever you actually want to target. So
All right, thank you. Thank you very much.
Yes, sir.
I think that is clear enough. Like those who are into phytomedicine who are screening for novel compounds, what actually we can see they're looking for is these ligands that they can use to target specific maybe proteins or genes that are involved in some diseases of interest to them. Am I right?
Yes, sir. Yes, sir.
Thank you. Please, if you have question, you can signify by raising up your hand. There is, or maybe by typing it in the chat box there. Tim, there is one person there.
Yeah, there's a question.
Okay, you can read it and answer, please.
Okay. So it says, can you throw more light on gene finding database, the function and usefulness? And are these database specific for only human? Okay, so for gene database, if you look at this slide 3 that I shared, you'll see genomes. That's a database for gene. Then there's gene bank, there's gene 3D, then there's gene quiz. And it's not specific for only human. It could be for animals and human, and then even microbes. So it's not specific for only human. So the essence of these databases are, the big goal is human, right? You want to treat something in human, probably a disease in human, but they might have worked on this particular gene in a lower organism. It could be microorganism, it could be maybe lower animals and all of that, which is easier to study than having to study human gene. So a whole human genome is kind of like quite difficult to study, but they must have done this in probably microbes or smaller animals. So you can use this because they are always like a little similar. Not a little similarity. You could see some even 99% similarities to human. So when you do the alignments of all of these organisms, then you come up with your profile of similarities, and then you take the ones that is high ranked. So that can still be like linked to like the human genome. And that makes sense because we also do experiments with mouses and mice and several, even rabbits, or some people use microorganisms or smaller organisms. So, and it makes sense because you have to progress. It's a progression before you get to like preclinical trial or clinical trial. So you are progressing from there. And if you see that, okay, this works, then it could be approved to the stage where you get to clinical trial and drug discovery eventually. So I hope that answers that question. So for the gene databases, they are not specific to only human. So you have the genome, the gene 3D, gene bank, gene quiz, and several other databases for gene study. Thank you.
Thank you very much. Are there other questions? There are two, I can see two messages here. There are no new questions, right?
There's a question from.
Can you read it, please?
Okay, there's another question that says, please, could you bring more light on docking definition and the role in drug development? Okay, yeah, so we have another session that will deal with this, but I'll just give a slight introduction to docking. So docking, like the word means, so just as I was saying before, that you have a target and then you have a small molecule. So you want to see how they both interact with each other. Your target, which could be a protein, could be a gene, and then you have a small molecule. You want to see how this interacts or how this can affect the activity of this, and then you join it together. So that's simply, in simple terms, that's what docking does. So you are bringing those two molecules together to form a complex, and then you want to see how this activity or the joining, so in layman's language, how this joining will affect the interaction of your big picture, which is your target. So you have a small molecule, it could be, there are several sites. So I think we also have a lecture that we explained that. So you could, in a protein, you have an active site, you have allosteric site, and then you could have like a binding pocket for probably other molecules and all of that. So if this ligand is docked to probably the active site, what effects will it have on the target? Is it going to increase its activity? Is it going to reduce or inhibit its activity? Or will it have no activity at all, no interaction at all? So you want to keep trying. And there are also several tools to do that. You can use your sitemap to be able to get your best fits for the protein molecule where the ligand can be docked. So docking is just like marrying something together or joining things together. So with the sitemap, you can be able to like predict where will be the best fit for the small molecule. So if we'll go into that later in the lecture, you'll see probably the induced fit techniques, the lock and key techniques. So all of these are like basic things that you've been taught in enzymology, or one way or the other, you've had access to such information. But docking is just like a cliche or a word for just joining, that's bringing two things together or forming complex. I hope that explains what docking is.
All right. Thank you very much. I believe that is clear. If there are no further questions, we'll move to the next lecture. But before then, there is an interesting paper that you need to read about Introduction to Bioinformatics. It has been posted to the link has been posted to the chat box there by Dr. Fatuki. So please, you can click the link, get the paper, and then read. Don't forget, we said you can't learn everything about bioinformatics in a day. But through this training, those who are even complete novices, they should be able to start out. And then those who have some knowledge before, they can get some improvements. The goal of the study is that by tomorrow, you should be able to do some molecular docking and bioinformatics work on your own. So thank you, Dr. Hannah. You still have some other lectures later on, so we thank you for the clear presentation. We move on to the next lecture. The moderator will introduce the next lecturer now.
Sorry, sir, there's actually a question that was just posted now.
All right, please, you can answer the question.
Okay, sir. So it says, so after docking, what next? Will that lead to a drug discovery? So the big goal is drug discovery, right? So you don't just want to dock and stop there. So the scope of this training is also supposed to help us through after docking what next. So the reason why you want to dock is to be able to get the interaction, how the small molecule interacts with your targets. And you could do that through probably molecular dynamics simulations and several other computational tools, several other computational analysis, to help you with how the ligand molecule affects your targets. So then with this computational model just shorten, just as I said before, that if you want to use the experimental, it could take like 13 years before you could actually get to preclinical level, and then your screening and all that. But with computational models, you'll be able to shorten and then also to shorten or to limit the cost intensity. So with that knowledge that you have, you can then go to the experimental lab to begin to work with your targets at mind. So this has saved you having to like purify over 2,000 small molecules. So that, thank you.